In this question, you have to identify the correct sequence of events during mitosis. Now, the correct sequence of events during mitosis would be number 1, condensation of DNA so that chromosomes become visible during early to mid prophase. Number 2, nuclear membrane disassembly begins at late prophase or transition to metaphase. Number 3, arrangement of chromosomes at equator occurs during metaphase, which is known as congregation. Number 4, centromere divides or splitting occurs during anaphase, forming the daughter chromosomes. Number 5, segregation also occurs during anaphase as daughter chromosomes separate and move towards opposite poles. And number 6, telophase leads to formation of two daughter nuclei. So the correct sequence is condensation, then nuclear membrane disassembly, then arrangement at equator, centromere division, then segregation, and finally telophase. So the correct answer is option A. Now option B is incorrect because crossing over does not occur in mitosis. Rather, it is a feature of prophase 1 of meiosis. Option C is incorrect because this omits the step of nuclear membrane disassembly, which is very essential for spindle attachment to chromosomes. Now, option D is also incorrect because again, crossing over does not occur in mitosis. Anaphase promoting complex, that is APC, is a protein degradation machinery that is necessary for proper mitosis of animal cells. Now, if APC is defective in human cell, which of the following is expected to occur? Now, APC is a protein complex that is necessary for separation of daughter chromosomes during anaphase. So, if APC is defective, then sister chromatids will remain attached and the chromosomes will fail to segregate or separate during anaphase. Hence, the correct answer is option B. In this question, you have to identify the stage during which separation of paired homologous chromosome begins. Option A, packaging. It is incorrect because it is a stage of prophase 1 of meiosis where homologous chromosomes are fully synapsed and crossing over, that is the exchange of genetic material, occurs. Option B, diplotin. Now, diplotin is the stage when paired homologous chromosomes begin to separate. And this separation occurs due to the dissolution of the synaptonymal complex. But the homologous chromosomes remain connected at points which are known as chiasmata. Hence, it is the correct option. Option C, zygotin. It is also incorrect because it is a stage where homologous chromosomes begin to pair up, that is, synapsis occurs and the formation of synaptonymal complex also occurs. Option D, diakinesis. It is also incorrect. It is the final stage of prophase 1 where the homologous chromosomes are fully condensed and chiasmata move towards the end of the chromosomes, that is, terminalization takes place. Cells in G0 phase. Option A, exit the cell cycle. Now, cells in G0 phase have exited the cell cycle, and this phase is the resting state where cells are not actively dividing and they have left the cell cycle of growth and division. So many differentiated cells like the neurons enter G0 and may remain there permanently. Hence, this is the correct option. Option B, entered the cell cycle. It is incorrect. Now, cells in G0 phase are not actively entering the cell cycle. Instead, They are in a state of quiescence. Now, while some G0 cells can re-enter the cell cycle, but under certain conditions, as this is not their primary state. Option C, suspend the cell cycle. This is also incorrect. While cells in G0 phase are not dividing, they have existed rather than suspended the cell cycle. A suspension would imply a temporary halt like a cell in the G1 checkpoint, whereas G0 is a distinct exit from the cell. Option D, terminate the cell cycle. It is also incorrect. Cells in G0 phase do not terminate the cell cycle permanently. They simply exit it temporarily or indefinitely. The cell cycle as a whole continues in other cells, so this is not a termination. In this question, you have to identify the correct sequence of the phases of the cell cycle. Now, the correct sequence of the phases of the cell cycle is G1, followed by S phase, followed by G2 phase, and followed by M phase. In G1 phase, that is the gap 1, the cell grows and synthesizes proteins and organelles in preparation for DNA replication. Then in S phase, that is the synthesis phase, DNA replication occurs, resulting in the duplication of chromosomes. In the gap 2 phase, that is the G2 phase, the cell continues to grow, synthesizing proteins and making final preparations for mitosis. And in M phase, that is in the mitosis phase, the cell undergoes division where chromosomes are separated and cytokinesis occurs. Thus, the cell divides into two daughter cells. So the correct sequence is once again G1 to S to G2 to M. Thus, the correct answer is option D. In this question, you have to identify the correct statement with regard to G1 phase of the cell cycle. Option A, cell is metabolically active, grows, but does not replicate its DNA. Now G1 phase, that is the gap 1 phase, is the first phase of interphase which precedes DNA synthesis, that is the S phase. So in G1 phase, the cell prepares for DNA replication but does not actually replicate its DNA. Now instead, it synthesizes proteins, increases in size, and accumulates energy reserves in preparation for DNA synthesis in the subsequent S phase, that is, the cell is metabolically very active. Hence, it is the correct option. Option B, nuclear division takes place. It is incorrect. Nuclear division, whether in mitosis or meiosis, does not occur during the G1 phase. It happens during the M phase. Option C, DNA synthesis or replication takes place. It is also incorrect, as DNA synthesis occurs during the S phase and not G1 phase. Option D, reorganization of cell components takes place. It is also incorrect. The reorganization of cell components happens mainly during the M phase when the cell prepares for division. But G1 is more focused on growth and preparation for DNA synthesis. In this question, you have to identify at what phase of meiosis dissolution of the synaptonymal complex occurs. Option A, diplotin. The synaptonymal complex begins to dissolve during the diplotin stage of prophase 1 in meiosis. And this allows the homologous chromosome to start separating, but they remain connected at the chiasmata where crossing over has occurred. Hence, this is the correct option. Option B, leptotin. This is incorrect. In leptotin, chromosomes start condensing, but the synaptonymal complex is not yet formed. So the question of dissolution does not arise. Option C, packaging. This is also incorrect. In packaging, the synaptonymal complex is fully formed and crossing over or the genetic recombination occurs. Option D, zygote. This is also incorrect. In zygote, homologous chromosomes start pairing through a process called synapsis and the synaptonymal complex begins to form. So the question of dissolution at this stage does not arise. In this question, you have to match the components under column 1 with that of column 2. During the zygote stage of meiotic prophase 1, the homologous chromosomes pair up and become aligned side by side, and this process is known as synapsis. And the alignment of the homologous chromosomes is facilitated by the formation of the synaptonymal complex, which is a protein structure that holds the chromosomes together. Thus, A matches with 4. Now, in the packaging stage, crossing over or genetic recombination takes place that involves the exchange of genetic material between non-sister chromatids of homologous chromosomes. And this process results in the formation of chiasmata, which are visible points of contact between the paired chromosomes. Thus, B matches with 3. Now, during the diplotin stage, the synaptonymal complex begins to dissolve and the homologous chromosomes start to separate slightly while remaining connected at the chiasmata. Thus, C matches with 2. The dikinesis stage represents the completion of prophase 1 and terminalization occurs as the chiasmata move towards the end of the chromosomes, marking the final stage of genetic recombination. Thus, A matches with 1. Hence, the correct answer is option D. Some dividing cells exit the cell cycle and enter vegetative inactive stage, which is called quiescent stage, that is G0. And you have to identify this process occurs at the end of which phase. Now, the process where some dividing cells exit the cell cycle and enter a vegetative inactive stage, that is G0, occurs at the end of M phase and the beginning of the G1 phase. Now, during the quiescent stage, cells remain metabolically active but do not proliferate unless the organism requires it. And this stage is reversible. That is, it allows the cell to maintain a basal metabolic rate as well as transcriptional activity, and it can enter the cell cycle whenever it is required to. For example, the budding yeast Saccharomyces cerevisiae enters this G0 when it runs out of essential nutrients, and it enters the cell cycle upon getting the favorable condition. Hence, the correct answer is option C, that is, this process occurs at the end of M phase and beginning of G1 phase.